Ay, sorry. Sorry. Sorry sa mga online. Ha? Ang online? Ang online, it's your turn daw to shine. Sayaw na po daw mo. Nakaano pa sila. So again, after knowing the history, the different botanical and various crafts ways of classification, now we are going to proceed with where are we now in the Philippines in terms of crop production and agricultural production, not only crops. So in terms of una-una, our economy is being measured by GDP, gross domestic and product. So just a sum up of everything, although di man yun siya nag-change. I already have downloaded I also have a separate, ipakita ko mamaya. Crop statistics. Sa kadagan kasi din ako makita ba na. Manufacturing. Ito ara. Hindi na ko siya makita ay kalagot. Try na ako dari sa website. Start. Mali siya yung pagka. Anyway, pwede naman to siya i-search natin. Na mali kung save ba sa mga folder. Gani, ang crop statistics sa Philippines. So just to sum up, now in terms of wood, our top market and top export like coconut oil man yung top export na to. And which is 42%, followed by banana and pineapple. And our top country na for export is Netherlands, Japan, and US. In terms naman of imports, so kaninang wrap up lang if I'm going to show you the crop statistics, it's too much to bear. But kuha na to ang points but sometimes yung pinaka-details na very the least expected, yun pa yung lalabas. For example, it's not on my time, but in some other, I have heard na like how many daw ang mga staff sa DA in aning region. Which is very specific and very detailed na siya ng mga questions, but somehow mugawas po siya, no? But kung nagtrabaho ka sa DA, you have an idea, di ba? So like for example, kasi taga-DA ka rin. So sa inyong palang daan sa department ninyo, mga pila man mga staff? Sa region. Dagan po, no? So at least we have an idea. And depende po na sa choices. And in terms of top imports, ang ginapalit nato from other countries are actually top is cereals, wheat, no, and hindi maklaro, but it's so hindi siya maklaro, pero nakalimot ko ka, but something na manon na siya sa ano, ganyan, hindi siya maklaro. Naka-clipper kasi ko. And equal lang na ako siya, but kaya naman ko ng another sa na presentation for this. And our top imports, followed by soybean and milk. No? Top major supplier naman natin ay all from US. And in terms of farmers, asa mas daghan, lalaki, babae? For 77%. In terms of age, unsa po ang age range? It's not yet in here, but right now, nasa age 50, 40 up na ang mga farmers na to. No? And in terms of development, 56%. These are the different shares of the different crops, no? Ay, oh. Ay. Sorry. So we were actually discussing yung top major exports at imports in the Philippines. So export natin is coconut oil followed by banana and pineapple. While import natin is cereals, soybean, and milk. Nakita na karon, tanaw daw. Okay na? Makita na? So from the second quarter of 2021, ang performance ng agriculture is quiet. 2022 naman pa ngayon. And still, yung nakapublish na crop statistics natin ay nasa 2020 pa naman. No? So, but kanang ginapahimong question sa board exam actually is like mga at least 1 year na sila behind. Kasi hindi na siya updated pa kasi dugay yun ang processing. So as you can see sa animal na per sector, as you can see, ang pinaka 56% is from crops, then 14% livestock, poultry, and fishery 16%. So as of now, we have a total land area of almost 30 million hectares. 41% is devoted to agriculture. And then, kasi, diba, ang wage rate ng farmers is very low. Average na siya. And the average age of Filipino farmers is nasa 57. Diba? More retirable na sila, but wala yung retirement kasi ang being farmer. So our contribution to GDP is only 10%. 59 services, 59% is nasa services of food industry and everything. In terms of employment din, nasa 27% ang capability, ang ating portion of employment. Ganun talaga ka-importante po ang agriculture. So ito na yung ating performance as of up to 2016. So is it from 2014 to 2016? It is actually declining, diba? In fairness, nagot ako sa food. So we are already in here. So just in case, no, itatanong yung ang mandate ng ating agriculture and the vision and as well as the mission. No. So can I request Bonnie to read please the mandate? Mandate is responsible for the promotion of agricultural development by providing the policy framework, logic assessment, and business needed for the benefit of export-oriented. Okay, and our vision is, Sir Dre, please. Thank you. And our mission, Ma'am Bim? Okay, so yun yung ating vision, mission, and mandate. So are we doing it already? Yes. So, but eventually, no? And the DA banner programs as of this, this decade is of course for rice, corn, high value crops, fisheries, livestock, and organic agriculture. So the DA takes into account the value change also, no, in the implementing programs of the sector. So as you can see, isa sa mga kanilang mga priorities is the, hindi ko alam kung saan siya ka-view. It's from farm to table. Of course, you are very familiar already of this, no? For the steps, no? Till soil, plant seed, energy farm, crop, grow crops, harvest, processing hanggang transportation to wholesaling and consumption, waste management, and compost, no? Or composting or scrap. So, ito lang yung some of the intervention done by the Department of Agriculture in terms of providing seeds, farming tools, machines, and mga breeder stocks and livestock. These are just some. And also in the post-harvest processing facilities. And also research, training, and extension. Farmer field schools, no? Isa-isa yan sa mga extension program nila. And in terms of marketing assistance, by promoting local products and domestic and international markets. Ito yung mga example ng mga products. Linking farmers with mga buyers, companies. So in the marketing assistance also, they help the upgrading of standards. By doing Philippine national standards and establishing enterprises among each region. So dapat naka HCCCP siya And gap, so what do we mean by gap? How about HACCP? Hazard analysis, critical control point in post-harvest. And of course, halal, it means non-pork. So they have the so-called rising stars, or these are the crops that have gained local international attention. And these are being promoted by the DA. And they have potential for local market with nutritional value and potential ng mga uses. So example ay yung mga year-long rice sa Cordillera region. No, ayun yung mga varieties, cacao in all the regions. And coffee also. Adlay. Calamansi. Pili nuts. Okay, kamyas. These are some of the so as we all know, we are really very rich in natural resources. And we should, however, yung focus ng market natin is yung ano yung in higher demand, yun yung pinoproduce ng ating farmers. No, we don't know that the potential, like for example, yung ating malunggay or yung mangosteen capsule, diba? We are new to those production which have very high potential actually, even for export. Like yung okra, ginagamit yan sa Japan as anti-cancer. But we are not even producing them. In Thailand, they are already producing them in hectarage or large areas. So these are the following interventions they have, including local and international standard compliance, agribusiness promotion, trainings, post-harvest facilities, and trainings of farmers. So dati pa yan kay dating Secretary Pinyon. So have we changed so far? So again, di naman to kahit klaro kanina, no? But these were our top crop commodities: export, banana, pineapple, and tobacco, rice, and corn. And imports natin, tobacco, coffee, and rubber. So as the second quarter of 2021, actually, however, nag-decrease tayo ng negative 1.5. Crops, although have increased, but the livestock is very low. No? Poultry nag-increase, but the fisheries decrease in production. As well, in terms of current prices and growth rate. Current prices, ito yung mga current prices. Second quarter of 2021. Kasi wala pa silang report for 2022. No? And as of January 2021, ito yung mga different livestock. No? As you can see, wala may kabot of 1%, no? But sa Carabao, less pagani. Negative 0.4 pagani sa chicken. Now, I guess the next focus natin is those are just mga very specific na. So, ang dapat lang natin alamin actually, yung sino ba yung mga top producer ng region, kasi those will be asked in the board exam. No? In terms of the highest growth rate, ito overall ito eh. So, we will check one by one by ano naman siya, by crop. Okay, so I will not present this one by one kasi hindi na kasi to updated. Hindi ko na update, sorry. But I'll be giving you the summary of everything. So in terms of banana, pomelo, durian, sino yung nangunguna? It's but mango and tamarind, it's Ilocos region or region 1. And pineapple naman sa northern Mindanao. Ang calamansi ay sa region 4B. So I'll be providing you naman later the presentation. No? And ito summary lang to. Lanzones sa R and as well as mangosteen. Papaya sa Toxergen. Rambutan naman ay from Calabarzon and orange in Bicol region. Mandarin in Cagayan Valley and watermelon in Western Visayas. In terms of vegetables, eggplant, tomato, and garlic is from Ilocos region kasi sila yung pinakbet, di ba? Bitters. Then lettuce and ginger is from northern Mindanao. And usually yung mga high-value crops natin na mga cucurbits, not cucurbits, brassicacea. We have cabbage, broccoli, carrots, habichuelas, and Chinese pecha are all in the Cordillera Administrative Region. And asparagus, syempre, it's only from Toxergen. Ang palaya, okra, string beans, century corn, as well as onion. And gourd, radish, Calabarzon, ginger squash, Cagayan Valley, kangkong, arm, native pechay, Bicol region. Syempre, when we say rice, it's really it's the highest producer around Central Luzon. When we say corn, Cagayan Valley, cassava, doon sa, yes, arm. Ang sige ng arm? Autonomous region. Barna. Okay. And Mongo, peanut, Ilocos region, white potato, Davao region, and I guess from dito sa Digos, no? Kapatagan. Ube, Central Visayas, kamote, gabi, Eastern Visayas. Del Sur, no? Sa Mount Apo banda. In terms of cut flowers, usually Cordillera, Cardona siya, no? Gladiolus, rose sa may Baguio banda. Chrysanthemum anthurium, but orchids is from Bicol region. In terms of plantation crops naman, we have coconut, cacao, Davao region, of course, sa atin yan, coffee, cotton, and also banana. Coconut, cacao, and banana, coffee and cotton from Toxergen, sugarcane in Western Visayas, abaca, Bicol region, rubber sa Zamboanga, and tobacco, Ilocos region, kasoy, Mimaropa. So those are major productions and top producers of bawat region. And we will go back after this on the specific crops at saan ko ba yung kinuha. So please also remember our National Commodity Research Center. This also might be asked in the board exam and already, although like sa first topic na to, ang pwedeng lalabas lang is nasa 2, 3, no? And so what is CRDI? But I'm not really updated right now kung ano, kasi nag-change na rin. But still, we have the PTRTC, no? Philippine Tobacco Research and Training Center. Kasi kahit yung PhilPIDA, diba? PhilPIDA na siya ngayon. And Forest Products Research and Development Institute. These are the different research centers. And of course, Philippine Rice Research Institute. And we are actually very blessed to have the International Rice Research Institute in the Philippines. So, to continue, we also have the IPB, or Institute of Plant Breeding, the National Crop Protection Center, NCPC. These are all in UPLB. The National Plant Genetic Resources Laboratory, NPGRL, Farming System and Soils Research Institute, Post-Harvest Horticulture Training and Research Center, PHTRC, So internationally, we have the ERI, as I have said, in UPLB, Philippines. No? And we also have international now, Centro Internacional de Neuromariente de Matis et Rigo. No? And Centro Internacional de Patatas. Natay mga inanak, no? Those are just few. And right now, nana po yung mga bago-bago, but those are mga famous. No? And in the region, do we have in Davao a research center in here? Aside, of course, in the Bureau of Plant BPI and RCTC, no? Regional. So do we have a research center yet in the region? Agri-extension asa? Dere sa Intan. So, good thing that we have now. Bago lang na siya? 2019. Ano yun? I-take mo, ikon sa COVID siya? Agri-extension. Okay. Since 2019 na siya, no? Kasi, actually, all national research centers in the Philippines are based mostly sa, of course, main sa Manila, sa Luzon. And where actually the region is capable of. And we are hoping, the PBJA is hoping that we could have the banana research center here. Because we own it, diba? The banana industry, we should have it here, no? Not somewhere else. But until now, wala pa rin tayong banana research center. Kasi it has to be done. It has to be, sa mga Congress, no? I-propose na sa Congress, and an organization, an academe should propose for that, ask for the Congress help, whatever, mga political kaya. And then para gawin na siyang Republic Act, no? Kasi naka-Republic Act talaga yan. It takes process. And once naging research center na yan, then kasi it will be funded by the government. So all our taxes goes, somehow part of it will go there. And to give you finally, yung ating, kung saan ko kinuha yung mga statistic, ginagoogle naman ako na, no? So, Philippine Statistic Authority lang naman siya, no? Ayan, yung latest nila ay ang 2021 gipublish, but this is from 2016 to 2020 pa. No, you can just download it. So, as you can see, dito natin malalaman. So, 2016 to 2020, ano yung mga top production, top producers. So let me proceed to the highlights, no? In terms of cereals, so ito yung kanyang production. Volume of production. So in terms, this is million tons per hectare. So dapat may idea tayo, ha? Like you can even use this for your research or in reference to your own production. Like when we say in the rice or the palay production in the Philippines as of ito ibig sabihin area harvested, no? So nasa hectares ni. Ang production is this one. So 19 million hectare lang. So this one is metric million tons. Dapat metric tons. Okay, in terms of million tons, we have 19, no? But this is in terms of million hectare kasi. Dati kasi ang data is naka per hectare. So madali lang siyang i-convert, no? So more on irrigated tayo than rain-fed. So the top producer, ito na, as of 2021, pero yung binigay ko sa inyo, yun yung summary ng mga top export, top imports. So in terms of palay, we have, of course, hindi na natin isali yung ating other region kasi marami silang bumuo dyan. So still, we have the Central Luzon. Okay. And in terms of area, volume yun yung, ha? Of course, we should be expecting again the Central Luzon, followed by Western Visayas, Cagayan Valley, and so on. Maklaro ba yan? And for irrigated, kasi rain-fed yun, still Central Luzon, no? As well as in terms of distribution or area. Rain-fed palay by region. Distribution is in Western Visayas na yung highest. So sa corn naman, we have 8. So far, hindi naman siya decrease or increase siya. We have 8 million metric tons, million metric tons. 8 million metric tons and 2 million hectare. But the highest compare from are the yellow corn. Ito yung ating for feeds, then the white corn. So ang highest natin, producer of corn, in terms of area is yung Cagayan Valley. How about in Cagayan Valley pa din? In terms of white corn is Northern Mindanao na siya. And followed by carbs. So sa Mindanao lang kusog og production sa white corn kumpara sa yellow corn. And Cagayan Valley din in terms of distribution. So sa ating vegetable crop, banana, you can see here Davao region, no? Followed by Northern Mindanao and Sorsogon. But that is for distribution or area. And in terms of varieties being produced, of course, mas madami pa yung Cavendish, followed by Sab-A and Lakatan. So this is for overall bearing calamansi, no? So dapat alam natin yung how much and how large. Nasa 8,000 hectares na 3 tons, and also number kasi bearing area planted and production. So may maropa, no? Pag calamansi, may maropa. So yun lang ang dapat ninyong always tandaan, or if possible, i-memorize ninyo which area, ano yung top producer na region, ano yung crop. So yun yung mga tanong sa board exam. And also yung mga, not so much kasi sa area, buti na lang, no, sa yung mga data on how much and the volume of production in the area. So for manga, it's the Ilocos region. Usually naman by year, ang volume nag-change. At saka ang area hindi masyado. But ang top producer hindi masyado nag-change over 5 years or 10 years. So fix yan siya. Although iba-iba yung report. 2020 pa kasi nila statistic. Yeah, in terms of course, we have Carabao mango, main variety. So sa pineapple naman, malaki yung production natin, no? Its area planted is 66 million metric tons and 2.7 million metric tons, and yung ating area planted is nasa 66,000 hectare. No, kasi baka itanong how much area of production for pineapple in the Philippines. So you have an idea. Don't worry kasi naman ay choices. So makita niyo mo sa choices. Pero basic ang choices is 65, 66, and 67. So dole na kayo siya, no? So nasa 66 siya in 2020. So top producer pa rin ang Northern Mindanao. So I'm just showing you, pero itong naka-list mo na to siya actually. Para dali na lang i-review ninyo next time for our post test. So do you have any questions or clarification? It's all in here, including mga non-food industrial crops like abaca. So these are 124 pages. Usually money ang mga lessons natin sa crop production and trade. Ang palaya fruit. Wala mo ito ganina, no? So, pag pinakbet gani, Central Luzon gina siya, no? Ng mga gulay. But when we say mga high value crops na vegetable, it is carb talaga. Pag mga lagot mo naman, na-deriva na sa atong arm, dyan sa Northern Mindanao. But for pineapple, is it Northern or Southern Mindanao? Northern. O talong na pod, Ilocos na po na siya. And Mongo, Ilocos pa din. So this is Central Luzon pod ang onion. Okay, so potato. Car. So, di ba kanina, double region to, ang white potato. So, as of 2020, it's car. Wala na siguro na-produce sa Kapatagan or Maragusan. So, i-update na lang ninyo dere, no? Okay, so kasi we have 1 hour and a half left. So, sorry ha, kasi I have to leave early at 4 o'clock. Okay lang. Then, overtime lang pa next Saturday. Until kahit hanggang 9 to 10 p.m. Joke ra. So much more on pre-production and mag-quiz, short quiz lang taana next Saturday para and also the scientific name para makakanta po ng uban. Ansa? Quiz lang siya. Good. Mga 100 items na quiz. Diba, Sir Dre, pila ganit, ay, hindi man dito si Golgi akong pa-historia. O si Sir JP. Kung gamay lang siguro ako ang i-post test, mga 1,000 items kada. So, kasi, ganit, oo. Ano sa inyo ni Sir Vincent sa inyo? Ay, kasi, kasi ang actual practice talaga is in the test. No? Kasi kani, kapuyan ta, malimtam na po nato, pag human ani, limata niya, pila pa kabulan. But I tell you, focus lang ta ha. Kasi pila ka bulan gud nga, bago ka mautong. Murag mga anak, diba? Kanang conjugate dinalandala sa imong tiyan. Pag man nag-exam, balag kalimtan na ninyo may tanan. Appeal kami, kalimtan na ninyo mga scientific day. Basta i-remember lang din na siya. During today hanggang sa day sa inyong exam. No? Pwede na mo masigo na una sa inyo ha, mga uyab. So we'll proceed with the plant anatomy and structures. So these are actually mostly videos ang presentation ako, but the one that I'm going to send to you in the GC are yung handouts ko na, ha, and PowerPoint.
Hi, it's Mr. Anderson, and in this podcast, I'm going to take you on a tour of the cell. We're going to talk about the
tour of the cell.
So here's the basics. Okay, so you have the flowers of the plant, the stem, you have the leaves, you have fruits, and then you have roots. And so let's talk about the roots first. So the function of roots is really two things. It's going to absorb water and minerals, and it helps anchor the plant to the ground on the right. And so there's kind of two different kinds of root systems. So root systems, this is a fibrous root system. There's no main root, like things like here. They're made for fast water absorption, and they help hold the soil and prevent erosion. These are found in monocots and seedless vascular plants. Okay, so like the firm. Then the other type of root system is taproot system. It has one main root, and then it has smaller lateral secondary roots. Okay, and And they have more stronger anchors that help absorb deep water. This is found in gymnosperm and diatoms. And now plants can modify their roots. They can have things like prop roots that help hold them up and support. This is actually a horn. They can use their supports for climbing, or their roots for climbing and support. They can have roots that are aquatic and help absorb water from a lily. They can modify their roots for storage and food storage, like a potato or that's a beet. So it's kind of an example of those. Now plants, now let's look at their stems. Okay, so in a herbaceous plant stem, herbaceous stem is all green, or you can have a woody stem that's got a wood in it. And that's the terminal bud at the top. Okay, and then it has the axillary buds on the side. The terminal buds are the bottom. Then it has what's called lenticule, which are the little bulky things on the wood. It has a leaf scar with a leaf biting up. And then it has a terminal bud scar where last year's growth occurred. Okay, so from you can see it's from one terminal growth to the next terminal growth scar is where 1 year's growth occurred. Okay. Plants can also modify their stems, just like they modify their roots into thorns for protection. Think like cactus. Okay. They can modify them into tubers for storage, just like a white potato. They can modify them into stolons that actually is a type of asexual reproduction. It's a strawberry plant that's budding off. They can modify them into things like what's called the form, which is a storage. The onion is a modified stem. They can modify them into tendrils that are used for climbing and support, which is this is a vine. Let's look at their leaves now. So you can see all the different parts of the leaves, and you're going to be labeling your lab activities. Obviously, if it has parallel leaves, it is a monocot. If it has netted leaves, it is a dicot.
Mutation, no?
Leaves, it's one leaf, where they can have a compound. And that entire thing from where it says see the petiole and all those little leaflets, that's actually one leaf. Where there's like 6 or 7 leaves on one, it's actually 6 or 7 leaflets on one leaf. Okay. And then the monocot, you can see all the different parts of the monocot and the dicot. That's in your picture. So you can look at the monocot versus dicot in all their pictures. So you can see things like a monocot has a fibrous root system, and a dicot has a taproot system. And a monocot has parallel veins, and a dicot has netted veins. And a monocot, you guys know, has flowers in parts of 3s and 4s, and a dicot has flowers in parts of 4s. This is something new. Monocot has vascular bundles that are scattered. Those are the little parts that are these little things. These are the vascular bundles. Okay, so that's where the xylem and phloem is. Whereas vascular bundles are more of a ring structure. You can see these little this going around the ring. This is each of the vascular bundles in a dicot. Okay. Plant, now we're going to look at some thin out. We're going to start to get inside a little bit more. So in plants tissues, they have a different types of tissues, just like you have different types of tissues, muscles and epithelial and all that kind of stuff. The first bit of tissue for a plant is they have what's called ground tissue. And they have, and ground tissue can be made out of parenchyma cells, which are thin walls used for storage and photosynthesis. And they can also secrete things. Those are what you call the typical plant cells. Another type of ground tissue is collincula. Collincula have thick, flexible walls used for support, and they're usually found in young shoots. Okay, so young shoots, young stems. Sclerenchyma, our last one, they also have thick walls, and they're used for support, and they are found where no growth is. So it's kind of like what's left behind. Kind of no longer there. You can also, plants also have dermal tissue. They have ground tissue, they have dermal tissue, which is like the epidermis, this covering surface, okay? Think that is the guard cells, that's what that pink thing is going to, okay? So those are the outer cells. And they're gonna do protection and water absorption. Water absorption. Okay. Then you have another type of tissue is you can have vascular tissue. This is xylem, okay? And so xylem is going to transport water and minerals. It's made out of tracheates, which are really long, thin cells with tapered ends. You can see those ones on the right. And then it's also made through vessel elements. And the vessel elements are shorter, wider, and they're also fatter. So you can see that in the fatter cells here, that would be a vessel element, and then these would be tracheates if you're looking down on them. But either way, they're big fat cells with really pretty big walls. And they're all dead, dead cells. Vascular tissue that's phloem, a little bit smaller, and it's obviously transporting food. And they have 2 types. They have sieve tube members and companion cells. When we get into how the phloem actually works later this next semester, very beginning of next semester, you're going to see why those are important. Just know there's two different things. So cells can grow in two different ways. They have primary growth, which is growth in length. And that happens at what's called the apical meristem. And it happens at the tips of the roots and the tips of the shoots. Okay, you can see that's where lots of mitosis is occurring in that case. And then there's secondary growth, which is growth in width. And that's where you have lateral meristem. And there's the vascular cambium and the cork cambium. So the vascular cambium and the cork cambium. We'll look at those parts in a minute. The cork cambium is going to be more on the outside. In the root tip, though, you have what's called the first area is you have the root cap, which protects the apical meristem. Then the next area is going to be the zone of cell division. That's where actual cell, that's the apical meristem. That's where the cells are dividing, mitosis is happening, things are occurring. Okay. Then you can have the zone of elongation. That's where cells are going to absorb water, they're going to elongate, they're going to get longer. Okay, they're going to start to grow. And then you have the zone of maturation. So you have those 4 zones, and they go. That's where cells are maturing. They're going to start to perform specific functions. They're going to start to differentiate. You're going to get some root hairs. All those things are going to happen in that area. This is a herbaceous dicot stem. So I'm going to highlight that because this is not a tree. Think it's like a green plant, a green stem. So this is what it looks like. That's got its epidermis, its outer layer of cells, and it provides protection. And then inside, is the cortex where it's pointing there. Okay, just right there. It's made out of ground tissue, and there might be chloroplast, which is green. And then there's pith in the middle, and there's storage area, and then it provides support through like trigger pressure. Okay, so just think like lots of osmosis happening there, and then there's the vascular bundle, which is the xylem and phloem, and it occurs in the rings structure. Remember, because this is a dicot, so it's going to occur in the ring structure. Now we're going to zoom in on one of these. Okay, one of these vascular bundles there. So the vascular bundle. So we zoomed in on one of them. Has the bundle cap with cells that are dead, fibrous, and thick-walled, big fat cells. Okay, and then these are mostly sclerotic bundles, old cells, and then they're going to help provide support. Okay. And then you also, in your vascular bundle, you're going to have your phloem, which transports food, with sieve tube members. They're long living cells. And then you're also going to have your companion cells that are smaller parenchyma cells. And then you're also going to have your xylem. Those are your big cells transporting water. They're dead tracheids that are dead with their big wall. And then you have your vessel elements that are larger in diameter, and they're also dead. And then finally, you have your vascular cambium, which is between the xylem and the phloem, and that's where cells are dividing to make for your secondary growth to make the xylem and the phloem. Okay. Now let's look at a woody stem. So in the center, so this is a picture of a tree. Okay, in the center you have your pith. Then you have a lot of xylem, which is your wood. Okay, and you can have summer wood, which are smaller cells because there's less water available. And you can have your spring wood, which is where there's larger cells, more water available. And you can see the annual ring. There's the first year's growth, and then the second year's growth, and then the third year's growth. Okay, that's the secondary growth. And then you have your bark, where there's phloem, and then there's a cortex. Okay, and so that's kind of where the outside part is. And in between the xylem and phloem, just like in a herbaceous dicot stem, you have your vascular cambium. That's in your xylem and phloem. That's going to help produce new cells, the secondary growth. And then now we're going to look at some roots. Okay, so here's a dicot root cross section. So if you're looking at a dicot root, that's the epidermis on the outside. It's going to absorb the water. There's root hairs to help increase the surface area for water absorption. And it also helps provide protection in the epidermis. Then you're going to have your cortex. That's where all the storage of starch occurs. That's what all those little purple things are. And then you have your vascular cylinder, or also known as your stelae. And that's where you have your xylem and phloem. Okay, and then there's an endodermis that outlines it to help protect it. We'll talk about how that works. Why that's important when we look at water transport and things. And so we're going to zoom in on your vascular cylinder. There's your xylem, your big fat cells, large dead cells, transport water and minerals. And then there's your vascular cylinder parts that are phloem. Okay, and they're small dead cells that transport food. And then you have your endodermis with your Casparian strip that's waxy and helps form a barrier. It helps regulate what goes through. So everything has to go through the endodermis or that Casparian strip before entering the stelae, before entering the vascular cylinders. It can kind of help regulate it. It's kind of like a larger cell membrane. And then up at the top, okay, last thing, we're gonna look at the leaf here, okay? Because up at the top of the tree, there's the upper epidermis, which provides protection. And it has a cuticle that is waxy and it decreases the water loss, okay? And then on the lower epidermis, that's where you have your stomata, those little holes that allow carbon dioxide in and water and oxygen out, so because photosynthesis is occurring. And they're surrounded by guard cells, the cells that protect them and help open and close them. And then you have palisade mesophyll, which is made out of parenchyma cells. They're very tightly packed cells, and that's where lots of photosynthesis is occurring. And then you can have spongy mesophyll, where there's more parenchyma cells that are loosely arranged, and there's more photosynthesis is occurring. And there's air spaces to help with gas exchange for photosynthesis. And then finally, you have your vascular bundle, or these are also known as veins at this point, because you have your xylem, your bigger cells, and your foam, which is green. And then there's a bundle sheath cells surrounding the bundles that help kind of protect it. And that's all. I know it's a lot and I talked fast because there is a lot, but just really just kind of go through, go back over it, look at the pictures again. You have all the pictures. We're going to do a lab with it. It's really just you guys got to learn where things are and what they do. That's kind of it.
O, diba? Paspas kayo siya kasi there are a lot of things daw. So, although we have limited time, but as an exercise, we'll have another exercise again. And syempre, masayo na dito mukanta ang PLD, no? So we'll have 3 groups. The first and second, third group, no? So you will, you will work as a group, and then you will just mention, syempre, maulan niya, second and third. Mag-mention, kabangan mo, no? Pero meron ka na ganyan, hindi nga mo, hindi ka mo, nakakanta sila, but in this case, mention ako sa kapal, for example, a tower for production of seeds. Yeah. Nagkaroon ng grupo ng bean, so there's what for diversion of water. And the next one was needs for the photosynthesis. So naman ninyo, one part, one function, one part, the next function, and then ano siya ba? Kasi ay wait ko yun. Kung isa lang ang sukuan, mastorya, kung na-estorya dayon, nakapamulyo na nakaimay. Pero di na pwede na siya mausap na pag-leggas o pwede sa pagkakala. Hindi na siya ubulan na yun at pinagod mo, there are thousands of plants, there are 6 main parts of the plant, from roots and one flower. Like flower. Kasi there are thousands. So ready na mo? Isang naman Pinoy ko ay. Electron Acceptor. Malaya na lang daw sila, Carlo. Just any parts lang yun sa kuan, sa plants, no? And their function. And sorry ang mga online ha, stay put lang mo. Okay, ready na mo? Ayun lang kayo, seryoso, ha? Ha? So, ang kanina sa post-test, ang ginagamit natin is positive reward. Maging sa'yo makatap mo ito tagaan of prizes na to. But during quizzes, negative reward. Makasayaw ang mapilde. So, pag-tikay lang ang istorya. Yeah, ready na ta? Di ba mo ka-memorize ang mga, cell wall, isa pa na. But this actually will come out in the board exam. Hindi na mawala ang plant anatomy and physiology functions. Lagi na siya. So, dapat kabalo mo ang psi functions sa mga different parts of the body. So, anong na root hairs? Actually, ang root hairs ang capable nga mag-absorb. So, just like mag-apply ka fertilizer nga nung around the canopy man of the fruit tree. Okay? Nagla ang mga fresh root hairs. No? It's the one who can absorb the nutrients in water. Or para dili na lang kayo lisod sa inyo ha. Parts na lang, walay function. Charo? Parts na lang, walay function. Sige, exercise naman na siya ba? So start ta sa group 3. Parts lang man, wala may function. Oo. Okay, ready? 1, 2, 3, go! Group 2. Group 3, 1. Stem, 3. 2. 1. 3. 2. 1. 3. 2. 1. 3. 2. Manalo. Alas, sana pang group 2. Sana pang group 2. Oh, sige. Naka-practice na ako kaya para Gino. Ano itong para Gino? Oh, stand up. Para G ka na. Kamo na lang. Oh, di ba? I-practice ay. Okay, stand up na. Group 2. Ansa? Kamu na lang kanta? Kanta para jipod ug sayaw. Sige, 1, 2, 3, go. Ay, with sounds talaga. Sige, sige. O, sakta, with sounds yun daw. Kani siya, no? Lain na po. Tapos siguro yun lang. Okay na eh? Kaya na ni mo na eh? Picture na niya. O, diba? Ha? Wala pa, oy. Nabili pa ko. So next na, after this one, let's go very specific. Sure. Different cell parts naman.
Hi, it's Mr. Anderson, and in this podcast, I'm going to take you on a tour of the cell. We're going to talk about the different types of cells and then how the structures inside a cell fit their function. First thing, though, that we need to talk about is why cells are small. The reason cells are small is that material moves into a cell through a process called diffusion. So oxygen gets in that way, and carbon dioxide is going to move out in the same way. And so it would take a long time for material to diffuse into a cell. And so what we can do is we can actually make that volume the same, but we can increase the surface area. And now the distance the material has to move is actually relatively small. And you also might think to yourself, well, why aren't they infinitely small? Why are they really, really tiny? Well, the reason why is that the material inside a cell, the information inside the cell, like the DNA and the machinery of the cell, has to be able to fit inside the cell. And so there's like a perfect sweet spot in size for all the different types of cells that we have. Another thing I want to talk about is cells are not boring. When I grew up, I had this idea that a cell was like a bag of jelly and you had stuff like a nucleus inside it and it would essentially float around. This is probably perpetuated by biology teachers always assigning like an edible cell assignment. And if you actually look inside a cell, it's incredibly complex. They have this cytoskeleton that's made up of a number of different macromolecules. It's like a lattice inside the cell. And all the organelles fit within that lattice, and it works almost like a monorail as material is moved around on this monorail using these motor proteins. And I'm not joking, they literally walk like that on the monorail. So they're incredibly complex, cells are, but they're oftentimes misunderstood. And they were totally invisible to scientists until we invented the microscope. In other words, we couldn't see them. If you look at your hand, you can't see the cells. And scientists couldn't see them either, so they didn't know what was going on until they discovered it and invented the microscope. Comes in two different types. You basically have optical microscopes and then electron microscopes. Optical microscopes use light and lenses to magnify the image. If you've ever used binoculars and then you turn it upside down and hold it close to your hand, you actually have a real simple microscope. And so that's the way that they work. If it's an electron microscope, what they're using is a number of magnets, and those magnets will be used to focus electrons, either through an image or bouncing it off an image. So we've got transmission and scanning electron microscopes. How does this work? Well, a quick demo would be to take a big magnet and hold it really close to an old television or your computer screen. Don't do this. If you were to do it, it would permanently ruin your monitor or your computer screen. But basically what it's doing is the magnet is changing the path of the electrons. And by doing that, we can actually increase the magnification on the specimen. So here's some pictures that were taken with these. This is the paramecium with an optical microscope, one that you have in a typical biology classroom. These ones are taken by a transmission electron microscope. These are little viruses, or this would be an ant that you're looking at. Now these two are dead because the material, in order to look at it, the process is actually going to destroy it. In fact, in here you have to put a thin layer of metal on it that we can bounce it off on a scanning electron microscope. And so the future is electron microscopes, but it's also what are called fluorescent optical microscopes. So we're coming up with these beautiful fluorescent dyes. You saw one on the first page in this podcast. and that we can stain material that can stay alive. I even saw one stain this last summer that was a live dead stain. And so you would stain it and it would show you all the cells that were alive at that point and dead at that point. It's really cool. We're getting some great visualizations. First thing you should know is there are two major types of cells. We have what are called prokaryotic cells and then eukaryotic cells. Prokaryotic cells are going to lack a nucleus. They're before the A if we break down that word. So there's going to be no nucleus. Eukaryotic cells are going to have a nucleus. What types of things are prokaryotic? Really only two things. Bacteria are going to be prokaryotic, and then the archaea bacteria, let me try to spell that correctly, are going to be prokaryotic. Eukaryotic are going to be things that you think of as alive that aren't microscopic. Things like plants, animals, fungus, protists, things like that that are really, really large. And the scale is bad here because if I were to scale it right, the protist, or excuse me, the bacteria would be about the size of this mitochondria. So these are really, really small, but there are some similarities between them. In other words, all cells are going to have nucleic material, so they're going to have DNA. All cells are going to have a cell membrane around the outside, some form of cytosol on the inside, and they're also going to have ribosomes. Now, they may differ, but all cells are going to have those things. As we move to eukaryotic cells, go back again, then we're going to have organelles. So we're going to have organs within the cell that you're familiar with, like a mitochondria. And so basically, prokaryotic cells are simpler. I'll talk more about them when I talk about bacteria, but most of the time in this podcast, I'm going to talk about eukaryotic cells. This would be an animal cell, I can tell right away. And so let's kind of look through an animal cell. So basically, these are the major organelles that are found within a cell, from the nucleus all the way down to the centriole. And so what I'm going to do is go through it, show you where they are, talk about what they do, and then you probably want to review at the end and go through all of them and see how much of the information you actually picked up. So let's start with number one, and that's the nucleolus. Nucleolus is going to be found within the nucleus. And I used to be confused on how this actually works. What they do is all the chromosomes that are within the nucleus, what they do is they put all of their genes to make ribosomes in one area within the nucleus. And that, as a result, since we have a lot of proteins inside here, is going to be a little darker when it gets stained. So this is an area where the chromosomes are all producing ribosomal RNA to make the ribosomes. It's going to be right there. It's kind of a two-step process. So basically what happens is in this area, they're going to produce ribosomal RNA, it'll roll out here to actually build some of the proteins using ribosomes outside of the cytoplasm. Then those proteins will move back where we assemble the building blocks of proteins, which are going to be ribosomes. So I talked about a lot of different things, but what did I mean to talk about? Well, the nucleolus is an area where the ribosomes are assembled inside the nucleus. If we go to the next one, the next one's going to be the nucleus, and that's one of the first organelles that was ever discovered. There's a beautiful fluorescent dye on the nuclei. So what's the function of the nucleus? Well, when I grew up, I always heard it was like the brain of the cell. That's really oversimplifying it. What's inside here? Basically, we've got DNA. So the genetic material of the cell is going to be found inside the nucleus. And that's going to determine, what kind of a cell it's going to become. But it also is going to control the cell. In other words, we're going to make proteins, we're going to make enzymes at a certain time. And as a result of that, cell's going to do something. So if you still want to think that it's the control center of the cell, that's okay. But a better way to think about it is just where the genetic material is. And it's also going to have little pores on the outside that'll become important when we start talking about transcription and translation. So there are going to be little holes on it. And that's how material can move out and material can move in through those little holes. Okay, next we get to the ribosome. Ribosome, generally growing up, I represented those as little dots inside the cell. It's a little more complex than that. The two parts of it, you're going to have a small subunit on the bottom, you're going to have a large subunit on the top, and the messenger RNA is going to move through that. And then on the top, we're going to bring in the transfer RNA, and we're actually going to build our protein on it. And so the function of the ribosome is going to be to build proteins. And prokaryotic and eukaryotic have different ribosomes, and that's how some of our antibiotics actually work. Vesicle is a broad term. Vesicle basically means a membrane-bound container. And they're really, really small, and sometimes they're really, really big. So a vacuole will be an example of a vesicle. And they move material around. Depending on what they do, like a transport vesicle will move material around. Next, we get to the level of the rough ER, the rough endoplasmic reticulum. It's actually a membrane that is continuous with the nucleus. And so we've got this folded membrane that comes out from the nucleus. You then have ribosomes that are sitting on the outside of it. And that's why it's called rough ER. I like to think of this as the factory inside of a cell. And so basically what you're going to have is this membrane. So you've got a membrane like this, and then you're just going to have a ribosome that sits on the top of it. So basically what you can do is as the messenger RNA comes through, we can make the proteins that we want to make. And so it's like a factory. It's going to be where we make the material. It also produced the membranes that are going to be used within the cell. Next, we get to the level of the Golgi body. I like to think it looks kind of like pita bread that is folded on top of itself. So if we were to say, where are these proteins going? They're going to be created in the endoplasmic reticulum. They'll then be packaged in a little transport vesicle and moved to the Golgi apparatus. At the Golgi apparatus, we're going to modify that. We're going to add things like carbohydrates to those proteins. We're going to snazz them up a little bit, and then we're going to send them on their way. So another way to think about that is it's like a UPS. In other words, it is a shipping part of the cell. Things come in as a transport vesicle, they're going to go out as a transport vesicle, and they're going to go where they need to go within the cell. Next, we got the cytoskeleton. Cytoskeleton is the structure inside the cell. It actually gives it that physical structure. As it moves, if a cell were to move around, that's going to have to be like an amoeba. That's going to do with the cytoskeleton as well. The way I like to think about this is through analogy. So it's kind of like a bridge. So on a bridge, you're going to have two things. Those are going to be supporting the bridge, but then you're going to have these really thin wires that attach it up, like on the Golden Gate Bridge. And so basically inside a cell, we have those two things. We have the big things. Those are called microtubules, and they're made from a protein called tubulin. And then we have these really thin things, and those are called microfilaments. And what the big things, the microtubules do, is they provide compressional support, just like the weight of the bridge is supported by them. And then those thin microfilaments are going to provide tensional support. And so if you think of a cell like the Golden Gate Bridge, but kind of inverted inside it, that's a good way to think about what a cytoskeleton is. Next, we get to the smooth ER. What's it missing? Ribosomes. What's it producing is going to produce a lot of the lipids, cholesterol, things like that in the cell. It also is really, really important in detoxification, so breaking down toxins. And so if you're an alcoholic, hopefully not, but if you're an alcoholic, basically, the more you drink, the more your body's going to build up smooth ER inside itself. So you're going to have to drink more and more and more. Next, we've got the mitochondria. Mitochondria, you know, is the area where we're going to generate energy. What's it really generating? That's going to be ATP in the form of ATP. It basically has a folded membrane inside a membrane. It looks a lot like a bacteria, and that's because scientists think that they became parts of our cells through endosymbiotic theory. In other words, they became parts of the cell. They produce ATP for that cell, and then they get a place to live. What's some evidence for that? Well, they have their own DNA. They produce on their own through binary fission. And so it's pretty much accepted as biological fact. Now we have the vacuole. Vacuole is going to be something that we find inside plants, not in animals generally, large vacuoles. And in this plant cell here, what it's doing is it's storing water. So it stores that balance and pressure, that turgor pressure that keeps the cell properly inflated. Some protists will actually have a contractile vacuole that can pump water out when they're living in a freshwater environment as well. We've got vacuoles, but they're really small in general in animals, and they're used for like endo and exocytosis. Next, we got the cytosol. Cytosol, you can think of as like the dissolved material. So it's the fluid, but it actually contains solutes inside it. We used to think that was about it, but what we're finding is there are concentration gradients within the cell. And so even the cytosol itself is pretty complex. Next, we go to the level of the lysosome. The lysosome is going to be, sometimes it's been coined as like the suicide sac. What does it really have inside it? It has these digestive enzymes inside it, and it's contained within this membrane. And so basically what we can do is we could have that go next to another vesicle that has material that we want to break down. And those digestive enzymes will go in there and it will break it down. Or where it gets its name from is if we were to pop this lysosome, basically what happens is those digestive enzymes would go throughout the cell and kill the cell, dissolve the cell. And so the process of apoptosis, where the cell kills itself, is a product of lysosomes. And finally, we have the centriole. Centriole is part of what's called the centrosome. And basically, it's important in positioning within the cell. So depending upon where the centriole is, it's also going to set up where's the nucleus going to be and where the other parts of the cell are going to be. It's also important as the cell divides, it's going to be, as it migrates to either side, it's going to initiate the formation of the spindle. And the spindle is going to be attached to the chromosomes and going to pull it to either side. And so we have those, but if you're looking at higher plants, they don't have centrioles and their role is somewhat undefined. And I think we could say the same thing for all of these, that we really have an idea of what they do, but they probably do lots of other things that we're really not familiar with. And so this is where the podcast becomes scary. I'm going to make all those terms disappear. And basically, if you hit pause, could you go through at the beginning and list, what is number 1? What is number 2? What is number 3? What is number 4? What does number 1 do? And if you can't do that, you really don't understand it. And working with kids in class, what I found that when you're trying to learn the parts of the cell, sometimes it's easier to just build some flashcards and go through the flashcards. Because if you can't get it right now, then you don't got it. And so that's a tour of the cell, and I hope it was fun, and I hope that was that.
Okay, so ready na mo sa rematch daw yung ni Madam Bim. Ha? Rematch daw the parts of the cell and its function. Parts of the cell lang. Gamay raman to siya. Diba, Sharo? So after na lang, it's either parts of the cell or function. Para mag-mind. Okay, na? Parts or function. Kasi, nilalang ko sa part na so, pero na, pag mausap mo tuloy mo, big over na, no? Ipamemorize na lang mga punta sa natin. Ready? It's either part or function. Para sa parts din. Pwede po kung isa ng part ng istorya, kung ang next function, yung part na sige. Next level na lang mo. So start ka sa number 1. 2. Nakilos lang sa number 3. 1. Superior. 2. Ribosome. 3. 1. Centriole. 2. Cuticle. 3.
Golgi Body.
1. 2. 3. 1. Inner membrane. 2. 1. Outer membrane. 2. Chicken dance daw. Ay, kuhaan man yung Dennis. Wala siya yung dance. Isa ka ba na sa kuhaan? Okay na yun, nice na yun. Sige na! Sabda. Bili mo, ane? Daghan mo niya siya. Android, ha? Unsa to? Android? Dalagang Pilipina na version? Muna yung pasabot? Dance. Ha? Android? Kana? Asan ni? Ano? Sige na, go na. Wait lang. Group ko last ba niya? Kani? Or kani? 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 Sa ubos? Sundugan ako ninyo.
The free version checks basic spelling and grammar. The premium, on the other hand,
Muna siya? Muna? Ang sapa? Kane? Madali na. Happy birthday, Sir Jeffy. That's why I'm talking to the girl in the mirror. Oh, wala pa daw. Balos na saan mo? So the next is after plant anatomy and physiology, we're going to discuss about photosynthesis and respiration. I know how na kayo muna, no? Asa? Hanap akong kuhain. Pag nawala akong photosynthesis, direct. Ano kasi wala siya dahil? Okay, so before tayo mag-photosynthesis,
Hi, it's Mr. Anderson, and in this podcast, I'm going to talk about plant nutrition and transport. I'm going to use a number of terms that you should be familiar with, like monocot, dicot, or ground tissue, vascular tissue. And if you're not sure what I'm talking about, make sure you go back and watch the previous podcast on plant structure. But before we get into that, we should talk about what plants need.
Wait lang ha. Diretso na ito para ano.
Vascular plants produce nutrients such as sucrose in their leaves. These nutrients must then be transported to the rest of the shoot or to the root tips where growth occurs. The leaves are referred to as the source, and the shoot and root tips are referred to as the sink. A source is an organ that produces more sugars than it requires, and a sink is an organ that consumes sugars for its own growth and storage. There are two kinds of vascular conducting tissues in plants: xylem, which conducts water and dissolved minerals, and phloem, which conducts carbohydrates, mainly sucrose. The transport of sugars throughout the plant, called translocation, is carried out by phloem cells. Phloem cells are connected to one another end to end by sieve plates. These are perforated structures that create a direct connection between the cytoplasm of connected phloem cells. Phloem loading is the process whereby carbohydrates enter the sieve tubes at the source. As a result of phloem loading, a high concentration of sugar develops in phloem cells near the source. Phloem loading results in a lowered water potential compared with adjacent xylem cells, causing water to move from xylem to phloem by osmosis. This influx of water creates a high turgor pressure near the source and lower turgor pressure near the sink, causing the movement of water and sugar from the source to the sink. Sucrose is removed at the sink by various active transport mechanisms depending on the cell type. In some plant cells, a transport protein located in the membrane of a storage vacuole called a tonoplast transports sucrose from the sink cell cytoplasm into the vacuole at the expense of ATP. This reduces the concentration of sucrose in the sink cell, which enhances the movement of sucrose from the phloem cells through the companion cells and into the sink cell. All these events produce a continuous flow of water from xylem to phloem and then back to xylem. Water leaving phloem in the root moves upward primarily due to transpiration, not due to water potential differences between xylem and phloem.
Okay, so as you can see the ending part, you should always remember ano yung function ng xylem and phloem, no? Xylem is the one, is the tube, okay, up inside the, and then it's through diffusion process and turgor pressure. For example, init kaayo, so mas kusog ang absorption na. Mas kusog ang transpiration, mas kusog po ang absorption sa roots, padulong sa taas. But when we say phloem, parang siya nag-flow and distribute sa mga carbohydrates, products of photosynthesis, and also water from xylem, no, to the different sink, di ba? Itong sink ganina asa ang parang ang needs growth and development. Usually ang mga shoot tips, mga growing flowers and fruits, those are the sink. So mas kusog ang sink dito pagpadulong ang mga carbohydrates or photosynthesis. So the key to actually remember all of this is to understand ang sa itong mga parts and parts sa mga parts of plants and the cells and their function. So, like, do we understand fully that the function of the cell? Then that's the time na we will remember that those vacuoles, no, nucleus, nucleolus, reticulum, ER, and rough ER, na. That's the time na ma-remember na to sila kung kabalo to sa function nila. I mean, function sa cell. So the cell itself, it is the basic unit ng life. Diha nag-produce tong ATP, genetic material, and so on. So I will not talk so much on that. And we'll proceed with tanaw ra pud ni. Daglimog ko kay nganong wala dali ako prepare gabi. So let's proceed with photosynthesis. Nagpila ganyan siya kakuan. So actually ang ako ma-take up lang diri is part 1 and part 5. The overview kasi para ni siyang 15 minutes na kasi siya.
Giving us background information, need for investigation and photosynthesis. Other videos in the series will take a closer look.
Enjoy daw kayo mo.
Phosphorylation of ATP can be osmosis, the Joven cycle, and the alternative pathways of carbon fixation as seen in
Di ang kay siya ko so.
So what do we know about how plants and other photosynthetic organisms make their food? What do plants need? We know that plants take in carbon dioxide. They need water. And they need energy in the form of light. They use these ingredients to produce sugars. So they liberate oxygen. So here's our overall chemical reaction for photosynthesis. But like we learned in our Introduction to Metabolism video, overall reactions don't tell the whole story. In fact, photosynthesis is a complex series of reactions that take place in 2 stages: the light-dependent reactions and the Calvin cycle or carbon fixation. So here's the short story. In the light reactions, we take in light and water. These are used to trap energy from the sun to form ATP and generate high-energy electrons. During the light reactions, water is split, which replenishes the electrons that are going to be lost out of the light reactions, creates hydrogen ions that we're going to use, and also liberates the oxygen that we see in the overall reaction. Now, the high-energy electrons that we make here in the light reactions, plus these hydrogen ions, get picked up by a molecule called NADP to form NADPH. This is an energy carrier for us. It's carrying those high-energy electrons and those hydrogens. And it's in the light reactions that we release oxygen. So in short, the light reactions take in light and water and produce ATP, a molecule called NADPH, which is carrying high-energy electrons and hydrogen, and oxygen, which is a waste product. Now let's move on to the Calvin cycle, or carbon fixation. So we take in carbon dioxide from the air, and then delivered to the Calvin cycle from the light reactions are high-energy electrons and hydrogens delivered by NADPH. Now we have all the products we need to make the glucose. We have carbons, oxygens, and hydrogens, and high-energy electrons to tie them together. But we need a power source. We need to add energy, and that energy comes from ATP. ATP is going to drive the reactions of the Calvin cycle.
We're going to use that energy in those reactions to rearrange these molecules into the high energy bonds of glucose. So we can summarize the Calvin cycle as taking in carbon dioxide and producing glucose, but it needs help from the light reactions. It needs those products from the light reactions, the energy delivered by ATP, and the high energy electrons to build these high energy bonds of glucose, and the hydrogens, because glucose is carbons, oxygens, and hydrogens. Now in our other videos, we're going to get into the details of how this is actually happening on the molecular level, at the cellular level, at the organelle level, what's actually going on in the light reactions, and the processes and the reactions of the Calvin cycle. But before we do that, in this video, we need to go over some of the bigger ideas first, some of the background information, and we need to talk about the nature of light. Now light is energy emitted as photons, which can act as both particle and waves. And since light is driving photosynthesis, we need to kind of investigate its nature a bit. What do we know about the speed of a wave? Well, we know that the velocity equals the wavelength times the frequency. So let's get a pen here. The velocity of a wave equals the wavelength, the lambda, times the frequency. But what do we know about the speed or velocity of light? Well, it occurs at the speed of light, which is approximately 671 million miles per hour. Well, so that equals, let me grab a pen again, that equals wavelength times frequency, because the speed of a wave is wavelength times frequency, so wavelength times frequency. But this is a constant. The speed of light is always the speed of light. It doesn't matter what color of light. In fact, all electromagnetic energy travels at the speed of light. So if this number is a constant, then what do we know about how wavelength and frequency are related? Well, hopefully you can understand that if this number can't change, that if wavelength goes up, frequency has to go down, or vice versa. As wavelength goes down, frequency is going to go up. So we can see that for light, wavelength and frequency are inversely proportional. When we're talking about light and its ability to help us make an energy-rich molecule, so we need to talk about the energy of light. Different wavelengths of light carry different amounts of energy. So let's look at how we understand that. We need to look at a concept called, equation called Planck's constant. Planck's constant is E equals hf, where E stands for energy, h is Planck's constant, and f is frequency. And again, since this number is a constant, we can see the relationship between energy and frequency, and therefore the relationship between energy and wavelength, so that as frequency goes up, and this is a constant, then energy would go up. But as frequency goes up, wavelength goes down. So we know that the shorter the wavelengths, the higher the energy, and the longer the wavelengths, the lower the energy, or vice versa, the higher the frequency, the higher the energy, and the lower the frequency, the lower the energy. We also know that different wavelengths of light equal different colors. When we look across the electromagnetic spectrum, when we talk about light, we talk about visible light, which goes from the reds to the violets. Roy G. Biv may be something that's familiar to you. And sunlight is a mixture of these different wavelengths. Now I'm talking about this because ultimately it's light that's going to be the driving force to add the energy into our system. The chemical bonds of glucose have high energy because they represent the absorbed energy of light. So how does a molecule absorb light energy? Let me back up and do that again. When a molecule gets struck by light, that photon of light can strike an electron and cause it to jump to a higher energy level. Now often when an electron gets boosted to a higher energy level, it immediately will drop back down and emit that energy back out in the form of either light or heat. The key for us is to try to trap the electron while it's at that higher energy state. So when light strikes the electron, knocks it up in energy, if we can grab it while it's at this energy state and put it into a bond at that point, at that state, that bond will have a higher energy to it than if we just use an electron that's back down at its resting state. Or I should say at its ground state. When sunlight hits plants, the energy of light is absorbed by the different pigments in the plant. Different pigments will absorb different wavelengths of light, aka different energies of light. If not absorbed, the wavelength is reflected. So I asked you this question: what color are most plants? Well, hopefully you came up with green, because chlorophyll is the major photosynthetic pigment. It's absorbing some wavelengths of light, but reflecting the green. So let's look at these graphs here that show, these are two different graphs kind of showing the same thing. I think maybe one's easier to understand than the other, that show the different wavelengths of light that are absorbed by the major photopigments found in plants. There's chlorophyll A, chlorophyll B, and the carotenoids, which are some of the orange and yellow pigments. We're going to focus mostly on chlorophyll, and in this graph they've kind of combined both chlorophyll A and chlorophyll B. And you can see that chlorophyll is absorbing the blues and the violets and the reds and the oranges, but reflecting a lot of the green, and so that's why plants look green. Now if we take a closer look at the structure of chlorophyll A and chlorophyll B, I'm not going to ask you to memorize this or even to recognize it, but what we need to understand is that these molecules are designed such that these pigments, that they're really sensitive to light. They can absorb light energy. So when light comes in and strikes this molecule, there are electrons in here that when they get struck by light can get boosted to a higher energy level. And again, the key for us is to grab this electron while it's at this high energy state. And these molecules are really good at donating these electrons to be boosted to a higher energy level. So where is the chlorophyll? We have to look deep down inside of a leaf. And if we look here at the structural diagram of a leaf, we can see that down in here in these mesophyll cells, these middle layer cells, you see all these little green dots. We need to zoom into one of these cells, and jam-packed in these cells are tons of these dark green circles, these organelles called chloroplasts. Let's take a closer look at a chloroplast. This is a neat diagram, kind of shows us an artist's rendition of a chloroplast, being able to look inside. But let's look at a diagram that that has labels on it. So we see the structure of a chloroplast. Chloroplast is a double membrane bound organelle. It's got an outer membrane and an inner membrane. And inside the chloroplast we have this system of stacks of membranes. These membranes are called thylakoid membranes. And the thylakoid membranes is where the light reactions are going to occur. And surrounding these stacks of thylakoids is a liquid area called the stroma. And the stroma contains the enzymes that are going to run the Calvin cycle. So we said that photosynthesis occurs in two distinct sets of reactions, the light reactions and the Calvin cycle. And if we look inside the chloroplast, we have two distinct regions, the thylakoid region and the liquid stroma surrounding it. But we need to take an even closer look. So let's go back up and look deeper into the chloroplast. In fact, let's zoom in on the thylakoid membrane. If we look in here, there are two structures that are very important to us, two more things we need to talk about. Photosystems, which are going to be our light harvesting complexes, where we're going to grab the light and absorb it. We have these clusters, whoops, I don't know why that keeps happening, these clusters of pigments, and also, let me make this bigger, electron transport chains, which we're going to use to tap into the energy of the high energy electrons. So let's get to it. What is a photosystem? A photosystem is a light harvesting complex. These are clusters of pigments that are embedded in the thylakoid membrane. When light strikes this photosystem, it can boost an electron to a higher energy level. This high energy electron, I've drawn red, then bounces around this photosystem. It gets passed from molecule to molecule and it bounces around until it reaches a special chlorophyll A called the reaction center. At the reaction center, the electron can get picked up by an electron acceptor, and so it's trapped at that high energy state. And then the electron acceptor is going to deliver the high energy electron to an electron transport chain. So it drops this electron off, and we'll talk about electron transport chains in just a minute. So again, let's back up and look at this one more time. Here's our photosystem. Light strikes the electron, boosts it to a higher energy level. Whoops, back that up again. Try that one more time. Boost it to a higher energy level. It bounces around the photosystem till it reaches the reaction center. When it reaches the reaction center, an electron acceptor can come in, pick it up, and deliver that electron to an ETC, or electron transport chain. Now when we do our details of light reactions, we're going to talk about two different photosystems. Photosystem 1, also known as P700, and photosystem 2, P680. And they have slightly different composition of photopigments, and so they kind of specialize in different wavelengths of light, and we'll talk about which different light reaction pathways that they're involved in. We'll get to that in our later video. But we need to talk about that electron transport. The photosystems were embedded in the thylakoid membrane, and so are the electron transport chains. Let's talk about what electron transport chain is. An electron transport chain is a series of enzymes embedded in the thylakoid membrane. Now, the electron acceptor from the photosystem delivers the high-energy electron to the ETC. The electron is dropped off. It's easy to think about this like a staircase, and the electron is going to move down the electron transport chain until it gets to the end. And when it gets to the end, it's going to become it's back to its ground state. So along the way, energy has been given off, and that energy is used to drive the production of ATP. So we can say that as the electron goes down the ETC, energy is given off to make ATP. But I put this in here. It says kind of, because there's a bigger story there. And if you recall, that kind is that it's not happening kind of directly like this. The energy that's given off as the electron goes down the electron transport chain actually drives the concentration gradient of hydrogen ions to one side of a membrane. And this is going to build up a gradient like water behind a dam, which will drive the chemiosmotic production of ATP. So now we have all of our background information. We know what plants take in, what they make, and what they give off. We know that photosynthesis takes place in two distinct reactions, the light reactions and the Calvin cycle. And we know that the product of the light reactions are important in driving the Calvin cycle. We know a bit about the nature of light, and we know that light energy can cause electrons to be boosted to a higher energy state, and we can grab those electrons, we can grab that energy. We know that chlorophyll is the primary photopigment found in plants, and we know that chlorophyll is good at absorbing that light energy and converting it to those electrons to a high energy state. We know that the chlorophyll is found in the deep part of the leaves inside the mesophyll in the chloroplast. We know that chloroplasts have two distinct regions, the thylakoid and the stroma, and that those two regions correspond to the two sets of reactions in photosynthesis. And we know that it's the photosystems, the clusters of pigments embedded in the thylakoid membrane, and the electron transport chains that are embedded in the thylakoid membrane that are really going to be where we're doing the work of the light reactions of photosynthesis. So check back for the videos on the specifics on these other parts of photosynthesis as we go into more detail on the hows of the light-dependent reaction, how ATP is actually made, the steps of the Calvin cycle, and other ways that we can grab carbon dioxide.
Oh, sorry na, ano na siya. So actually, there are different steps, no? Tatlo. The light reaction, phosphorylation, and the Calvin cycle. So it is where na the different ATP which we're going to be used during respiration, no? So we will skip on that kasi na overview naman siya, but the next video will be about the difference between the alternative
in our series on
photosynthesis, which is the C4 and CAM, no? Do you remember this types of plants? So there are 3 types of plants based on photosynthesis. No? We have the C3 plants. We have So wala po nag-break kasi mag-off pa ng So murag sobra na po ito nga break sa group to. So ang C3 plants, these were the one na merong normal na normal na mga DJ siya maklaro, wait lang. And then, normal nga photosynthesis. And most plants are C3, like the spinach, beet, potato, and so on. But C4 plants are, meron silang tinatawag na Kranz type anatomy. No? And these are, kanang naa sila, what was that? I forgot. Isa. Sige ito. Wala kasi po naka-state na rin, no? But nakalimutan ko. Anyways, C4 plants, they have the type anatomy na sila ay marag ka ng part na wala sa CT and CAM, which will be mentioned naman noon mamaya sa video. And CAM plants, mo to siyang succulents, orchids, and cactus, mga agave, which they are capable of conserving water, especially in the sa desierto. So the question is, all the rest will be mentioned naman sa video. But C4 plants actually come for crassulacean acid metabolism, ang CAM. And so, makalimot ko ba sa iyahang part, but they are called, ang sa nga part ang sa C4 plants. So they have actually developed just because of ng mga unfavorable condition. No? So, let me just go back again. Kasi naapoy mas very short lang dere. Respiration. Picture de gyapon ko. Okay. And so, follow-up question regarding sa ating video kanina on, so why does most of the color of the leaves are green? Bakit? Because of the chlorophyll. What does it does with the green color? Iyahang reflect instead na gina-absorb niya. Ang color na makita nato because of chlorophyll is green. And does photosynthesis occur without light? Yes or no? Naabay photosynthesis maski walay light. We all know kanina, it needs, of course, light para na yung energy katong dito na sa light harvesting, di ba? So, yes or no? Okay, initially, yes, it needs. But later on, after harvesting light, like what the CAM and C4 plants do, bundle sheet cells day akong ginanin. So, occurrence anatomy na siya bundle sheet cells na before siya go to Calvin cycle is ma-convert muna katong 6 carbon into para mas efficient siya in terms of photosynthesis. So C4 and CCAM are more efficient mas efficient sila, mas daghan sila og photosynthesis kumpara sa C3 normally. So like for example, yung pineapple, normally C3 na siya but eventually ang mga pineapple ay naging C4 na. So let's proceed with ang anak ko. Kapsay, yung video kanina. To talk about more of the difference.
Welcome to our fifth and final video in our series on photosynthesis. In this video, we're going to look at alternative pathways of carbon fixation, the C4 and CAM plants. Before looking at alternative pathways of carbon fixation, let's remind ourselves how carbon was fixed during the Calvin cycle. In the Calvin cycle, carbon dioxide was picked up by a molecule called RUBP, and when they combined, they created an unstable intermediate that immediately broke apart into two 3-carbon molecules called PGA, or phosphoglycerate. This type of carbon fixation, where carbon is picked up here by RUBP and fixed into a molecule of PGA, or 3-carbon molecule, these plants that do this are called C3 plants because the first stable intermediate has 3 carbon. Let's look at the structure of a leaf to see when this might be a problem. When we look at a leaf, we see that we have an upper epidermis, the middle layer, the mesophyll, where all the photosynthesis is going to occur, and a lower layer of epidermis. But at the top of this epidermis, plants produce a waxy coating called a cuticle. Now the benefit of this cuticle is that it helps prevent water loss from the leaves. Water has a hard time diffusing out across this waxy layer, so it helps the leaf keep their moisture in. And that's a good thing. But the downside is that the gases that we need for photosynthesis, the carbon dioxide that we got to get from the air, has a trouble getting through this waxy layer also. So the carbon dioxide can't get in. Conversely, the oxygen that's produced as a byproduct of the light reactions has a hard time getting out. It can't get through this layer either. So while the waxy cuticle is a benefit on one hand, it gets in the way on the other hand. So what's the solution? The solution are the stomata, these openings on the undersides of the leaves that can allow for the gases to be exchanged. So the the oxygen can diffuse out and carbon dioxide can diffuse in. And that's a good thing. But the bad thing is, even though we're letting the gases exchange, we're also going to lose water through the stomata, and that's just the cost of doing business. You know, we're not losing a lot of water across the surface, but we will lose water out of these stomata, and that becomes a problem in places where the conditions are very hot and dry. So, as a positive, the stomata allow for gases to exchange, but on the downside, it allows for water loss. So how do plants reduce this water loss? Well, one thing that they do is they close the stomata at night. And they also can close the stomata during the day at times where there's under heat stress. And so while the stomata are closed, something interesting happens. Let's start here with this as our starting condition. Notice we have a lot of carbon dioxide in the leaf that's moved in before we close the stomata. But now it's just gotten too hot. We've been losing too much water, so the plant decides to shut it down. The stomata close, trapping these gases inside. Now, as photosynthesis progresses, we're going to consume carbon dioxide and produce oxygen, and so the composition of the air inside the leaf is going to change. As photosynthesis continues to progress, the levels of O2 are going to keep rising, and the levels of carbon dioxide keep going down. So the question is, why is that a problem? Well, let's look back at carbon fixation. In the Calvin cycle, RuBP picks up carbon dioxide, but RuBP in a different environment where there's a lot of oxygen and not a lot of carbon dioxide, RuBP is not so picky. RuBP will combine just as readily with oxygen as it will with carbon dioxide. So instead of carbon fixation going on, we have a process called photorespiration. And photorespiration is when RuBP binds to the oxygen instead of the carbon dioxide. Now, this can drastically decrease the rate of of photosynthesis. Hold on, let's fix that. Here we go. If this occurs, this photorespiration, the rate of photosynthesis is going to drastically decline because RuBPs are bound to oxygen and are not available to bind to the carbon dioxide. And this can create a problem in many climates where the temperature is really hot and the air is really dry. So we need alternative mechanisms for carbon fixation. Specifically, we're going to talk about C4 and CAM plants. Let's start with a look at the C4 pathway. Now, the way to understand the C4 pathway is we have to understand that the C4 pathway relies on a very unique anatomical difference to ensure an optimal carbon fixation. Well, what do we mean by that? Let's look at a typical C3 leaf. We see the air spaces in here, the stomata and the air, and we see our mesophyll cells, and all of these cells have access to this air, access to any carbon dioxide and oxygen that may be in here. But when we compare that to a C4 plant, we see a very different anatomy. In the C4 plant, we have a layer of mesophyll cells that wrap around a layer of cells called the bundle sheath cells. And let me grab a pen here. These bundle sheath cells that are here, and surrounding them is, I'm going to grab a different color, these mesophyll cells. So we have this very unique anatomy. And in the middle of the bundle sheath cells are a vascular tissue, our xylem and phloem, that bring water up to the leaf and take the sugars away. So this difference in anatomy is going to be responsible for allowing what we're about to see happen. Let's take a closer look. In the C4 pathway, we're going to fix carbon twice, meaning we're going to grab carbon dioxide 2 different times. First, in the mesophyll cells out here, and second in the bundle sheath cells. And in doing so, the C4 pathway is going to keep oxygen away from RUBP. So remember, RUBP binds with carbon dioxide to fix it, but in an environment where there's lots of oxygen, we get photorespiration. So our solution to this is to not use RUBP. For carbon fixation in the mesophyll and the C4 pathway, we use a different molecule called PEP. PEP, notice that PEP will ignore the oxygen and go right to the carbon dioxide. PEP is more discriminate, it's picky. PEP, or phenylenol pyruvate, will bind to carbon dioxide but not to oxygen. So in the mesophyll cells, PEP will bind with carbon dioxide and convert to a molecule called oxaloacetate. Oxaloacetate is a 4-carbon molecule, 4 carbons, C4. The first intermediate made when we first fix carbon dioxide is a 4-carbon molecule in the C4 pathway. Let's look at it a little differently. Here we have a diagram showing a little bit of the anatomy and also a pathway. So here's our airspace inside the leaf. Here's our layer of mesophyll cells, underneath which are the bundle sheath cells, and underneath that we have our vascular bundle. So that's the anatomy. And notice the situation. We have lots of oxygen and only a little bit of carbon dioxide. So when oxygen diffuses in here, PEP ignores it. It doesn't bond. So it doesn't get picked up. But carbon dioxide, PEP has an affinity for, so it will bond. So when carbon dioxide diffuses in and gets picked up by PEP, we form oxaloacetate, C4. Then the oxaloacetate is converted to malate, another 4-carbon molecule. And here's where things get interesting. This malate diffuses out of the chlorophyll, out of the chloroplast, out of the mesophyll cell, and into a bundle sheath cell. And when it does, it splits into a molecule of carbon dioxide and a molecule of pyruvate. And waiting right there to pick up that carbon dioxide is RUBP. And it binds to carbon dioxide, fixing it a second time, and kicks it right into the Calvin cycle that we learned in our last video. The Calvin cycle, of course, I left out the details here.
At this
It's going to
point,
produce a sugar, a
yan yung
precursor
kailangan
sugar,
of
which then
sa
diffuses in the
Calvin
vascular
cycle, no
bundle
need
and can get
for
shipped to all parts
sunlight.
of the
So,
plant.
pwede na siya gabi.
If we come back up here, the
So,
pyruvate that we have here diffuses back into the mesophyll cells, and with a little help from ATP, gets converted back to PEP. So look at what's happening here. This top cell, we have 2 different cells involved in one metabolic pathway. The C4 pathway basically is a selective carbon dioxide pump. Even though there's a lot of oxygen and only a very little bit of carbon dioxide, it's going to ensure that that little bit of carbon dioxide that's here gets to the Calvin cycle, and it keeps oxygen away from RUBP, and it keeps photorespiration from occurring. Now you're going to have to go back over this and really look at the anatomy and get the steps of this, as it's not something that's straightforward, but take a look at how this picture relates to, let me back up here, to this picture and see if we can see where things are happening. And I'm going to grab a pen here. Let's see. So that first carbon fixation occurs here when carbon dioxide gets picked up by PEP in the mesophyll cells. Eventually we drop the carbon dioxide off here where it gets fixed a second time by RUBP. My pen's not working very well. And now let's take a quick look at cam plants. We're not going to spend much time here, but cam plants have evolved an additional or another pathway to fix carbon, and it's been in response to plants that face also very hot, dry conditions. So these plants, they keep their stomata closed during the day always. It's not just a sometimes thing. They're never going to have the stomata open during the day, and that's going to reduce water loss. But the problem is that really limits how they can photosynthesize because it's during the day that the light's out. So what the cam plants do is that during the night they collect carbon dioxide, they fix it and store it, fixed by PEP like C4 plants, and stored as malate. Then during the daytime they shut the stomata and convert the malate back to CO2 and begin the Calvin cycle. So they do photosynthesis in 2 stages. And here we can see a comparison of C3 or C4 and CAN plants. Notice the CAN plants do not involve two different cells. They don't rely on this unique anatomy. They rely on a unique metabolic pathway. They just have different enzymes involved in this process, but it's similar. Okay, so that does it on our video series on photosynthesis. Please go back and review all the videos, and let me know if you have any questions.
Okay, so ayun, no? Why is it called C4? Kasi it has a carbon 4. Unlike the C3, the usual photosynthesis process, it's only 3 carbon ang product, no, ang gigamit. And for CAM plants naman, it simply does, they only closes their stomata kasi sa arid region sa desierto, very init siya. So they just close their stomata and fix carbon dioxide during night. And that's how they do the photosynthesis. So since it's already 4, sorry, continue na lang natin yung respiration next meeting. So, do you have announcements, Sir Gab? Di ba ang kaumundang kaso ka ng gani, no? Tinduman at tasa ka ng mga staff ba? So, do you have any questions or clarification? So, hopefully, na-refresh atong mind about photosynthesis, no? Especially katong nag-graduate na dugay-dugay on biology subject. Kasi plant physiology and anatomy is really part of the board exam. So sa itong post-test, makita na ninyo. But the key is just to understand how, we don't even understand ourselves, no? Then how our process, sometimes we still go to the medical doctor na, noon sa day kung ano na, umubo na ko, no? But in plants, it's very easy to understand. So, and that way, ma-remember na to ang ilang function. So, nakay pangutana from the online and also sa inyo. Manghagit pa kag another round challenge. So, yun lang, no? Thank you very much. And also sa online. Sorry, di na ko magdugay, ha? But promise by next Saturday, mag-overtime na ta, maski paghantod 12:00. Charot lang. So, thank you everyone. Bye-bye sa online.